In this session, we're going to look at A2 gene technology. Okay, so the first part of this is looking at restriction enzymes, or how DNA is transferred or how DNA is obtained from a section of DNA. So that's using the restriction enzymes, which is essentially a pair of scissors which cuts DNA at a particular sequence. Okay, so if we watch this little animation.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Okay, so as this says, the restriction enzymes cut at certain recognition sites, as we can see here. So here's the same one as in the animation. So GAATTC, and you can see the anti-parallel strain to that is the same code running in the opposite direction. So it recognizes, this enzyme recognizes that sequence, and it will cut in that sort of stepped fashion, which means that it leaves these free bases, which are known as sticky ends. So as they talked about there, you could put something else, which comes from DNA from another organism, or some man-made DNA, as long as it's got these complement or sticky ends which are complementary to these free bases, then you can stick that in. So you can stick in other bits of DNA, and that's very important, as it said in genetic engineering. Other types of restriction enzymes will just have a plain cut or a straight cut, which doesn't leave sticky ends. Now, scientists can stick some bases onto the end of it to give it the same sticky ends as this, but this is just to show that you have different types of restriction enzymes. Okay, another way of obtaining DNA from a specific organism is using reverse transcriptase. Okay, so you hopefully remember this from AS. HIV virus is an example of a retrovirus. Okay, so it has RNA and the enzyme reverse transcriptase. And when it invades cells, the reverse transcriptase converts the RNA into DNA. Okay, and that DNA can then be converted into double-stranded DNA with DNA polymerase. That's another way of obtaining DNA. Okay, and you can see here this sort of step through. Here's your HIV virus. And it's got these different spikes on the outside, which it can use to attach to recognition sites on the cell. So that's the way of tricking its way into the cell. So then the the RNA is converted into DNA with the use of these restriction enzymes, which we can see here. Reverse transcriptase enzyme converts that RNA molecule into a single-stranded DNA molecule, or cDNA as it's known, and then that with DNA polymerase, which comes up in a second here, is then converted into double-stranded DNA. So these are other different types of things involved with the reaction as well. So then that DNA combines with the DNA of the organism, or the genome of the organism, and then that organism or that cell will start making copies of viruses or HIV viruses, okay? Or as it says there, viral DNA. So then with your endoplasmic reticulum, it will make certain lipids, and also proteins can be made in there as well. There's your different types of proteins, and that will then cause new viruses to be made from that type of cell. Okay. Another way of looking at that is the recent COVID pandemic, is that they have used messenger RNA and reverse transcriptase to, or within their vaccines. So they put messenger RNA and reverse transcriptase within the vaccine. That's given to a human. That human then makes, now it's not the whole virus that it makes, but it will make a certain part of that virus, for example, the spike. So the RNA is converted to DNA with the reverse transcriptase, which produces this spike within the body. Then your immune response produces antibodies to that spike, and that gives you immunity. That little slide there also shows similar types of things. So RNA used to make vaccines. Okay. Next bit of this is using a gene probe. A gene probe essentially is again a human piece of, or sort of man-made piece of DNA. So if we were wanting to see if someone had a gene causing cystic fibrosis, okay, we know that the gene sequence for cystic fibrosis is this. Okay, so we make something called a gene probe, which is complementary to that. So I would extract a cell from this person, use certain chemicals to break down the DNA from the cell, or extract the DNA and make it double-stranded. So break down the hydrogen bonds within the DNA. That makes it single-stranded. Okay, then I would make my gene probe. Now the gene probe would have to be complementary to this sequence of bases. So for C, we need 4 Gs. For A, it's 2 Ts, then an A, a C, then another A, then a T, then another G. So that's the first 10 bases there. We've made a gene probe, or we've made a piece of synthetic DNA which is complementary to those first 10 bases. Okay, so there's our gene probe. We allow that to combine with or put it into these bits of DNA. If it finds that particular sequence, then it will bind to it, okay, and make that double-stranded. And you can radioactively label that as well. And then when you take a look under the microscope, there you can see that person does in fact have that gene sequence. So they do have cystic fibrosis. Okay, so that can be used in a number of different ways. So your gene probe, here you can see, is your synthetic piece of DNA, complementary to a targeted gene. You can see the bases match there. It's also got a radioactive label, which means that you can see under a microscope. Okay, in terms of using gene probes, it can be used in gel electrophoresis and fluorescent labeling that you can see here. So here's a section of DNA. You use restriction enzymes to cut that up. You add your DNA fragments to a gel electrophoresis kit, which essentially passes some current across it, or voltage across it, electricity across it. That allows the fragments to move. Smaller fragments will move further. Longer fragments will not move just as far. You can then transfer that onto a membrane, use your gene probe, and it gives you that fluorescent band, which you can then identify as, again, somebody with diabetes or somebody with cystic fibrosis, etc. Okay, there's an example of gel electrophoresis and how it's carried out. So you put your DNA samples into here, you pass a current across it. Samples, based on their size, will move different distances, and then you can, as I say, use your gene probe to radioactively label certain parts of the DNA. Okay. Also, you can see here, this is how we amplify DNA. So produce new copies of certain bits of DNA, and this is something called polymerase chain reaction. Okay, so you have a small section of DNA. It could be that you've provided a sample. It could be that you've found a small speck of blood or a hair at a crime scene. Because you've only got a small amount of DNA, you can only carry out a certain amount of tests. So if you copy the DNA many times, then you can carry out any amount of tests on it. Okay, and the way that is done is you heat the DNA up first within your PCR machine, and that breaks down the bonds, the hydrogen bonds between the bases, which makes it single-stranded. Then you cool it down to about 40 or 60 degrees. You add primers or anneal primers to it. That essentially shows where you want it copied from. So it's a sort of starter point. Then you heat it up again in the presence of free nucleotides and DNA polymerase. That sticks the nucleotides to their complementary bases. So a bit like if you remember back to DNA replication. And then you have 2 copies of that DNA, which are exact copies. Okay, you do that over a number of times, and that makes many copies of your DNA. You can carry out all your different tests. Okay, they also talk about nucleotide sequences, okay, and short tandem repeats. So there's a short tandem repeat. So a lot of DNA is filler DNA, so it doesn't actually code for anything. So you can see here AGAC, AGAC. So that's repeated a number of times. That's your short tandem repeats, which fills up the DNA. This might be a coding strand or a particular gene for the DNA. Okay, and here you can see something called a single nucleotide polymorphism, or a change, or we would have looked at it in terms of mutations as well, where the original DNA sequence is AAACT, okay, which codes for that particular protein. Whereas if you have a single nucleotide polymorphism, so one of the bases on the coding strand would change. So rather than an A, you get a G. That codes for a different type of protein, perhaps, which then may lead to a different characteristic being expressed. Okay. Also talk here about gel electrophoresis and cutting up DNA fragments, as we talked about before. So the smaller fragments tend to move further within the gel. The larger fragments don't move just as far. Okay, and that can be seen using genetic fingerprinting. So again, very similar to the gel electrophoresis we looked at before. You have your restriction enzymes cut up the DNA. You put those, or you separate those by your electrophoresis. That can be treated then when it's single-stranded, and you can add DNA probes, etc., to it as well. And that gives you a single genetic fingerprint on an X-ray film, and those are individual for, or those are different for individuals because their DNA is different. Okay, and you can see here, this is your DNA fingerprint on the X-ray film that would be seen. Okay, the next bit's looking at gene transfer. So transferring donor DNA or genes into a recipient cell. One way that is done is by using bacterial plasmids. Okay, so bacterial plasmids is just a circular piece of double-stranded DNA. You can use a restriction enzyme to cut that up so it leaves your sticky ends. You can also cut a desired gene. So for example, an insulin producing gene is often talked about. So you take a piece of human DNA, you cut out the desired gene with the same restriction enzyme, it leaves the same sticky ends. So therefore it will fit within this gap within the plasmid. You put that plasmid back into a bacteria cell. You have a bacteria that, in the case of insulin, produces human insulin. Okay, so for example, here you treat your plasmid with your restriction enzyme, same with the human DNA. You add your ligase enzyme, which sticks it back together. You now have a plasmid which has got some human DNA into it, so it will produce, for example, that insulin gene. Okay, in terms of identifying which plasmid has the desired gene, we can use plasmids with antibacterial resistance. Okay, so here, for example, is a plasmid which contains resistance to 2 antibiotics, one called tetracycline and one called ampicillin. So antibiotics obviously kill off bacteria. So anything containing these 2 genes would not be killed off. Any bacteria containing these plasmids would not be killed off by ampicillin or tetracycline. Okay, now if we put our human gene or desired gene in between, in this case, the tetracycline resistant gene, that switches off the resistance to tetracycline. So anything with this plasmid would be resistant to ampicillin, but not resistant to tetracycline. Okay, so that what we can do there is you can put these colonies, which are these bacteria, which have these bits of DNA, or these plasmids, onto a petri dish which contains ampicillin. So you let those colonies grow. So you have a certain pattern, if you like, of colonies growing which contain the ampicillin resistant gene. If they don't contain that gene, they'll die off. So the ones that grow contain that ampicillin resistant gene. If you then transfer that or replica plate that, now for replica plating, essentially you can just use a little bit of cloth. You put it down on your master plate, and then you put it onto a replica plate, another plate with growth medium, and it gives you the same pattern. Okay, so here they've just replica plated this pattern onto another plate which has tetracycline antibiotic on it, and then some of the bacterial colonies will grow. If they do grow, then the tetracycline resistant gene is intact. So it would be one of these. The ones that die off would have this tetracycline resistant gene switched off. So it must contain some human DNA, okay, or DNA that you want to be picked up by the plasmid. So then you could identify that these colonies, so in other words, you'd have a look at this one, then you'd have a look at that one, and then that one and that one. Those are the ones you want. So they contain that recombinant DNA. Okay. And we can see a little bit of that in this next animation.
One of the first genetic engineering experiments was conducted by Stanley Cohen and Herbert Boyer in 1973. They showed that the gene for frog ribosomal RNA could be transferred and expressed in bacterial cells. The first step was to construct the plasmid vector called PSC-101. This constructed vector contained a single site for the restriction endonuclease EcoR1 and a gene for tetracycline resistance. The restriction endonuclease EcoR1 was used to cut the frog DNA into small fragments. Next, the frog DNA fragments were combined with the plasmid vector that had also been cut with EcoR1. The sticky ends of the DNA fragments aligned themselves and the segments were joined together using DNA ligase. Some plasmids incorporate genes other than the rRNA gene, and some do not incorporate new pieces of DNA. The plasmids were then transferred into a tetracycline-sensitive strain of Escherichia coli and plated onto a growth medium containing tetracycline. The cells that incorporated the plasmid carrying the tetracycline gene grow and form a colony of cells. Some of these colonies consist of cells that carry the frog ribosomal RNA gene. The researchers then tested the colonies that form after growth for the presence of frog ribosomal RNA.
Okay, so we can use that type of process to produce, as I say, human insulin. Okay, so the next bit, looking at genetically modified organisms. So obviously bacteria, you can take out its plasmid, as we talked about, you cut it up with a restriction enzyme. You take out, sorry, that's the human DNA. You take out your insulin producing gene. The plasmid would be removed, cut with the same restriction enzyme. you make your recombinant DNA and combine those together, identify that human insulin gene has been picked up, then you put that back into your bacteria, then you have a bacteria that makes human insulin, okay, and any amount of human insulin, okay, on vast scale, okay. Also, there are genetically modified plants. So with tomatoes, they're often known as the flavor savers tomatoes, and excuse me, with them, they have a piece of DNA which means that the tomato stays a bit riper, so therefore it's got an increased shelf life. Okay, and there are a number of examples in the textbook about this, but also the benefits of genetically modified crops. So they can be herbicide resistant, they can be pesticide or have pesticide properties as well. Certain disease resistant. There's your flavor saver, your golden rice as well as another example of enhanced nutritional benefits as well. Okay, so worth going over those. Also, in terms of genetically modified animals, they have this example in the book that gives different types of animals, worth knowing certainly some of these and how those are used. So sheep, for example, being used to produce clotting factor, which can be used to treat people with haemophilia who can't clot their blood. Okay, next part is looking at gene therapy. So gene therapy essentially means trying to either switch off or remove a defective gene within an organism. Okay, different types. You can use somatic cells, which are cells which can be removed from a person or an organism, or you can use germline cells, okay, which is before the organism develops. Okay, so essentially with your egg and sperm, and pass on modifications. Okay, so here you can see this is what we call a vector, which is a bit like a bus, which brings DNA from one place to another. So in this case, it's from outside a cell into inside the cell. So we tend to use viruses, or you can use viruses as vectors, so they transport bits of DNA and inject them into cells. Okay, so that would be trying to produce a new gene, or give the organism a new gene so it makes a particular protein. Okay, so adenoviruses, you can see here, a bit like your retroviruses, they can transport DNA. Retroviruses would transport the RNA, which is then converted into DNA. Those are some examples of organisms which are used. Okay, liposomes is another one that's just a piece of DNA which has that lipid vesicle around it, and it is able to get within the cells via endocytosis. Okay, cystic fibrosis is one example of a disease which can be partially cured, if you like, by gene therapy. So we use viruses. Sorry, with cystic fibrosis, individuals don't produce an enzyme which breaks down mucus in their lungs, so they tend to suffer from a lot of lung infections, etc. Their lungs don't work properly, essentially. So they have a gene missing. You can put that missing part of the DNA into a virus, inject that into, or transport that into cells within the lungs, and that will cause the lungs to produce that mucus enzyme, or mucus breaking down enzyme. Unfortunately, gene therapy is a partially cure, because every cell in the person's body would, or certainly in their lungs, would have that gene missing. So as the cells replicate, they will replicate, and more of these will replicate with the missing gene, so therefore it's only a partial cure. Okay. Then we go on and look at the Human Genome Project. So the Human Genome Project is a project that was set up, as you can see here, back early 1990. Okay, a lot of DNA, obviously, research was done before that. The Human Genome Project was to try and map out all of the genes within a human. Okay, it was completed in 2003. So we can see along here certain genes that were discovered, although it's a bit blurry, genes that were discovered at certain times within that period of between 1990 and 2003. So what we have now is that we know where genes that cause particular diseases. We know what chromosomes they're on, which particular part of the chromosome, and how that is passed on to different organisms. Okay. Then we look at microarray technology. Okay, so microarray technology is looking at these sort of different colored wells, if you like. And with that, what we do is we cut up normal cells. Again, you can culture those. You can fluorescently label those with gene probes. You add those to your little microarray trays. And when they change color, you can see if it has normal cells or if it has disease cells, etc. And it gives you this type of thing here, the different colors, which can then be used to show how genes are expressed. Okay, so it could be used to detect if people have certain diseases, etc. It's a bit like adding a gene probe, and then the gene probe changes color. Certain sections of DNA would cause that change in color, so it can be used to detect perhaps certain diseases, etc. Okay. Next bit of this really then is looking at genetic engineering, which is what we talked about there, and the benefits of genetic engineering and genetic modification. So we can see things like gene therapy, things like producing vaccines, etc., things like being able to produce medicines, etc., that we talked about, or insulin, etc., things like that as well. Okay. Then they talk about mice in terms of knock-in mice and knockout mice. So something that has been knocked in, it's switching on the piece of DNA or adding a piece of DNA. Something which is a knockout is then removing or making a piece of DNA inactive or not inoperable, as they talk about, so switching it off. So you can switch on certain genes, but also switch them off. So here you can see a knock-in is you're inserting a gene or switching a gene on. Knockout is removing that gene, or you can switch it off. Okay. Then they talk about these biohazard labs and why we need biohazard labs when carrying out genetic modifications. So obviously, if these genetically modified organisms get out of the lab, then that could cause problems in the wider community. So a lot of these tests, a lot of these experimental techniques are done under very high safety levels. So with these labs, you can see a lot of PPE here. You can wash in, wash out. Everything's done in very controlled conditions to keep it as safe as possible. So these would be your sort of purely or purpose-built labs where experiments can be done. Okay. Obviously, there are then ethical questions around gene technology and genetic engineering, etc., and also with genetic screening. So you can be screened for particular diseases, which you can use your DNA probes that we talked about before for detection of certain diseases. Obviously, there are questions then as to what that or how that information is shared, or who is allowed to carry out or be screened. What type of information do we screen for, etc. Okay, so do we pass that on to insurance companies, life insurance companies, etc. if you know that you have Huntington's disease, for example, which you develop in your 40s. Okay, so ethical questions around that. If ethical issues again with gene therapy, who decides what uses are good and bad? Should it only be available to people who can afford it? What do we what types of genes do we try to switch on or switch off, if you like, or replace in terms of gene therapy as well? A number of questions there you can see. Okay. Obviously, there are a lot of benefits to gene therapy and genetic modification in terms of medicines. Okay, so medicines can be made more personalized, okay, which we can see here. So normally you develop a medicine or a drug, it has a good effect on some people, no effect on others, may have bad effect on some other people, whereas in the future, if the medicine is more personalized, you could take samples of blood, person's DNA, you take tissue samples, etc., you link that with particular drugs that have been produced or particular treatments, then hopefully it has a benefit for everyone. Okay, and then the final part of this is looking at biobanks. So biobanks essentially are big storage facilities where people's DNA can be extracted and then stored, and then certain tests carried out on those gives us a better idea then perhaps things that can be used in the future. Tests can be carried out, genetic engineering could be carried out on particular pieces of DNA, etc. So that's essentially a biobank is a big store of people's DNA. Okay, hopefully that helped with the gene technology topic.